Hello, Mr. Ellman here, and subject for today's video is the central dogma. A lot of what we're going to do today is quite technical. It involves a lot of different processes, a lot of names of some different parts within the cell, and you will want to take this slow, so feel free to watch it multiple times, to pause, to take notes, and certainly look at the other resources that I posted on my website, so that if I don't explain it well enough for you, you can find someone else who will be able to do it. In terms of some of the things that you need to learn through this video and the text and the activities we'll do in class. Certainly, what is the structure and function of DNA? And important here is what is the relationship between a gene on DNA and a protein. Transcription and translation are going to be those processes that takes the information in DNA, converts that into an intermediate form called mRNA, and then translates that mRNA into a chain of amino acids that will then form a protein. So I'm going to skip ahead to the next section of the Prezi, which is available on my website, to RNA and how RNA is going to be transcribed from DNA. So the process here going from DNA to RNA, before we get into the details for that, we should take a step back and look at generally what it is that this whole series of processes involve. We have here on the right, this is a strand of DNA. DNA is going to be divided into different genes. Not all of DNA contains genes, but genes are just certain regions on the DNA molecule that are going to be transcribed. So if we take, for example, gene 3 right here, it contains a set of DNA. DNA has these two strands. Remember, they are anti-parallel, so we have from the 3' to 5' and 5' to 3'. It has that sequence of T's, G's, C's, and A's. The process of transcription is going to make a copy of mRNA that is complementary to the DNA, meaning that it is going to look very similar. It's going to match up with the sequence of DNA nucleotides, but with the one difference of replacing those thymine bases with uracil bases. So in the process of transcription, really 3 steps: initiation, elongation, termination. This is the process of starting a segment of RNA, making it longer, and then finally ending the process. RNA polymerase is an important enzyme here. The job of RNA polymerase is to make RNA. It's an enzyme that is going to physically attach together RNA nucleotides in a string and piece those together to make a chain. RNA polymerase is going to find a promoter. The promoter region is just the sequence of DNA that is a little bit before the actual gene sequence begins, and it has some type of noticeable sequence, something like TA, that repeats and that lets the cell know that the gene is about to begin. A transcription factor is going to be proteins that are going to help the RNA polymerase to attach to the actual DNA. And the reason why transcription factors are needed is because the cell needs to regulate when certain proteins need to be made. Because you have every cell is capable of making every protein, but only some proteins are needed by skin cells versus, say, a nerve cell. And so these transcription factors are present to ensure that only the necessary proteins are going to be made, and only at the right times. So here you can see this big blob is the RNA polymerase. RNA polymerase is attached to the DNA with those transcription factors at the place where the RNA needs to start. What will happen is that RNA production is going to happen in the 5' to 3' direction. So here you can see on the DNA, here is the 5' part of the DNA. So the 2 strands of DNA are going to separate a little bit. Here you can see here's the 5' end of the newly made RNA. And so as we build, here's the 5' end, and it goes towards the 3' end, adding on more bases. RNA polymerase is the enzyme. That enzyme is adding on new bases onto that chain. The DNA obviously needs to separate a little bit in order for this to be able to happen. When the DNA shows an adenine, RNA polymerase will add uracil. When the DNA has guanine, RNA polymerase will add cytosine, cytosine with guanine, thymine with adenine. And so this is the complementary part to it. Again, it's not a copy. It's not DNA is adenine and RNA is then adenine. It's adding the bases that match with it. So when DNA has one type, the base that matches up with that based on the normal base pairing rules, that is what's going to get added onto the chain. This process will continue down the length of the gene. Once you get to the end of the gene, then transcription is going to stop. The RNA is going to detach from the DNA, and that RNA is going to then leave the nucleus because in our cells, again, the ribosome is the place where proteins are made, and so the RNA strand is going to transmit. It's going to transmit the information from the nucleus out to the cell where the protein is physically going to be made. In terms of RNA, lots of types, more than 10, and each of those has very different functions, and some of those are well known and some of them are pretty complex. 3 types that are significant to our processes here. The first is mRNA, or messenger RNA, and this is the type of RNA that I just described being made. The mRNA carries the DNA, carries information from the DNA out to the ribosome. The transfer RNA, or tRNA, is going to bring amino acids to the mRNA at the ribosome, and that those amino acids will be used to make the protein. Ribosomal RNA, rRNA, those are the type of RNA that physically makes up the structure of the ribosome. So the ribosome itself is made up of a type of RNA. Now in eukaryotic cells, when that copy that we just described is made, the mRNA often needs to be processed, needs to be fixed up a little bit before it's actually sent to the ribosome. Now, a lot of this, again, this happens only in eukaryotic cells. It doesn't happen in bacteria, but we have things like add-ons. So we add on a cap and a tail onto that piece of mRNA. The mRNA is going to be analyzed by some other proteins to look for things called exons. Exons are not really supposed to be part of the gene. They have kind of extra DNA bases. Those things need to be cut out before the RNA is sent out to the ribosome. And then finally, we have these sections of DNA that are between genes. So we can have a gene. We can have a gene, and then we have some space, and then another gene, and then some space. And there is a lot of alternative theories about why these extra pieces of DNA are there, since they don't seem to have any obvious purpose. But a lot of that is simply just not necessarily understanding quite well enough what's happening within those cells during these processes, so we can't really know for sure why it is that that DNA is there. But certainly a lot of studies being done on it currently. So that's the process of transcription. That brings us over to the process of translation, which takes the RNA and turns it into a polypeptide, which will then be folded into that protein. So I need to cover what it is that the ribosome is made of, a little bit about the genetic code that's used, and then again that 3-step process to be able to make the protein itself. So this piece of RNA in red here in the middle, that's what was made through transcription. Translation is going to use that information taking bases in sets of 3 and using those sets of 3 to determine which amino acid comes next. So for example, UGG is going to be a code that is translated into the amino acid tryptophan. UUU for mRNA will be translated into the amino acid of phenylalanine. And so going down the line in sets of 3, this RNA information is used to build a correct amino acid chain. The ribosome is where protein synthesis happens. All cells, prokaryotic, eukaryotic, have this. 2 subunits, and the RNA is kind of going to fit right in between those two, where those 2 subunits are, below the larger subunit and above the small one. 3 sites on here, the A, the P, the E. The mRNA kind of goes from right to left based on this picture, starting at the A, moving to the P, and then finally to the E And so you could think of it as like a string moving from right to left through the ribosome. tRNA, you can see the structure here. It's kind of a folded piece of RNA, and at the folded end is where the amino acid itself is going to be attached. The bottom of the tRNA has is going to match up with the mRNA. The top of the tRNA is where the amino acid is going to be. In terms of the code here, the genetic code is universal. So this box right here that we'll get to and we'll use, that you certainly will have used in class, is going to be the same whether it is a very primitive bacteria or a plant or a fungus. These kind of triplet codes called a codon are these sets of 3, and every possible set of 3 is found somewhere on this table. So there's 64 possible codons, but only 20 possible amino acids. So you can see that in many cases there are duplicates. You know, CUU, CUC, CUA, CUG. Those sets of 3 will all code for the same amino acid of leucine. Sometimes an amino acid might have only 1, like tryptophan, or it could have 4 or more possible codons. So the code is redundant. The code has a start and a stop to it. That will be important for translation, which we'll get to with those steps. Now, we'll talk a little bit about some of these proteins, actually. You can look back at those pictures when we do a molecular biology unit later in the year. So same 3 steps as transcription, starting with initiation, continuing to elongation, finally termination. So in initiation, this is what's going to start the process, and it's the special codon of AUG. So when AUG is read, that is the signal for the amino acid methionine. So mRNA comes in, coming from the right, attaches to the center, the P site on the ribosome. The tRNA that brings methionine, that will bind with the mRNA. And that starts the process here. Next thing that happens, so we have methionine here at the end, and then the mRNA is going to slide over. And so at the A site, whatever the tRNA that will match up, that's complementary to whatever codon is on the mRNA, that's going to come in, it's going to slide in there and bind with the RNA. Then what's going to happen is that you can see that this amino acid is going to form a new peptide bond with the previous amino acid along the line. So peptide bond extends, and you can see where there was 1, 2, 3, 4 amino acids. This 5th one comes along, and now we have a chain of 5 amino acids. The mRNA is going to slide over. When the mRNA slides over, the old tRNA that has lost its amino acid pops off, and a new tRNA comes into that A site, and then we continue building onto that chain. So when we step back there, we can see that as we continue this cycle, tRNA comes in, binds to the A site, new peptide bond forms. This gets repeated over and over again, 1,000, 10,000, 100,000 times, depending on the size of the protein. Translation is going to terminate. When one of 3 stop codons is reached. So UAG, UAA, or UGA, when that codon is read, there is going to be, instead of a tRNA, there's going to be a release factor that will clip the amino acid chain away from the tRNA, and that we now have a free polypeptide chain that will float away from the cell. Then the ribosome is going to disassemble, and it will reassemble and get ready to be reused to make a new protein. This process, transcription and translation, happens simultaneously in bacteria cells, but because they don't have a nucleus. So the DNA, the mRNA, and the ribosome can all physically touch each other, but in eukaryotes we separate that out into 2 separate processes. Transcription happens inside the nucleus, whereas translation happens outside the nucleus at a ribosome that might be anywhere in the cell. So in total, we can see transcription happens. We have RNA polymerase making a copy of mRNA based off that's complementary to the DNA. It's going to be kind of sliced up and stitched back together. It's going to leave the nucleus and head out to the ribosome. At the ribosome is where translation happens, where we're going to use the anticodons that are on tRNA, which bring in amino acids. Amino acids will build a growing peptide chain. That polypeptide chain, once it's disconnected, is going to then fold up into the shape so that the protein can then do its function. Okay, again, if you have any questions, feel free to email Ask Me in Class. But that is the process of transcription, translation, and protein synthesis all in one.